Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Hamdi bin Halim. I'm from 822228. The title that I choose for my video presentation is Mechanism of Salt Tolerance in Plant. Before I want to discuss and explain more detail about this mechanism, I want to give introduction of what is a salinity. Okay, salinity represents a major abiotic stress that has been associated with a significant economic impact due to reduced agriculture productivity. Most plants are sensitive to salt stress, and salinity can inhibit plant growth by triggering ionic toxicity, osmotic, and oxidative stress. Based on the responses to high concentration of salt, plants can be divided into two groups, which are halophyte and glycophyte. What is a halophyte and glycophyte? Halophyte are plants that exhibit high salt tolerance, allowing them to survive and live under extremely saline conditions. And glycophyte, they are sensitive plants and unable to grow under saline conditions. Salt tolerance. The mechanism of genetic control of salt tolerance in plants have not yet fully understood because of its complexity. There are in fact several genes controlling salinity tolerance in the different species whose effect interact strongly with environmental conditions. Thus, genetic variation can only be demonstrated indirectly by measuring the responses of different genotypes. Salinity tolerance may increase or decrease depending on the plant species or environmental factors. For some species, salt sensitivity may be greatest at germination, whereas for other species, sensitivity may increase during reproduction. Plants have evolved several mechanisms to acclimatize to salinity. It is possible to distinguish three types of plant response or tolerance, which are the tolerance to osmotic stress, the Na+ exclusion for leaf blades, and tissue tolerance. Okay, for first mechanism of salt tolerance is osmotic tolerance. The growth of salt stress plant is mostly limited by the osmotic effect of salinity, irrespective of the capacity to exclude salt that result in reduced growth rates and stomata conductance. In fact, osmotic tolerance involves the plant ability to tolerate the drop aspect of salinity stress and to maintain leaf expansion and stomata conductance. But if the accumulation of salt overcomes the toxic concentration, the old leaf die and the young leaf, no more supported by the export of photosynthesis, undergo a reduction of growth and new leaf production. For this reason, increased osmotic tolerance involves an increased ability to continue production and growth of new and greater leaf and higher stomata conductance. The resulting increased leaf area will be benefit only plants that have sufficient soil water, such as in irrigated food production system, where a supply of water is ensured, but could be undesirable in the water limited system. At the end, while the mechanism involved in osmotic tolerance related to stomata conductance, water ability, and therefore to photosynthetic capacity to sustain carbon skeleton production to meet the cell energy demands for growth have not been completely unraveled. It has been demonstrated that the plant response to the osmotic stress is independent of nutrient level in the growth medium. Okay, next, second mechanism is Na+ exclusions. In the majority of plant species grown under salinity, Na+ appear to reach a toxic concentration before Cl- does, and so most studies have concentrated on Na+ exclusion and the control of Na+ transport within the plants. Therefore, another essential mechanism of tolerance involves the ability to reduce the ionic stress on the plant by minimizing the amount of Na+ that accumulate in the cytosol of cell, particularly those in the transparent leaf. This process, as well as tissue tolerance, involve up and down regulation of the expression of specific ion channels and transporters, allowing the control of Na+ transport throughout the plants. Na+ exclusion from leaf is associated with salt tolerance in cereal crops including rice, durum wheat, red wheat, and barley. Exclusion of Na+ from the leaf is due to low net Na+ uptake by cell in the root cortex and the tight control of net loading of the xylem by parenchymal cell in the stem. Na+ exclusion by roots ensure that Na+ does not accumulate to toxic concentration within leaf blade. A failure in Na+ exclusion manifests its toxic effect after days or weeks depending on the species and causes premature death of older leaf. And lastly, third mechanism is the tissue tolerance. Tissue tolerance entails an increase of survival of old leaf. It requires compartmentalization of Na+ and Cl- at the cellular and intracellular level to avoid toxic concentration within the cytoplasm, especially in mesophyll cell in the leaf, and synthesis and accumulation of compatible solute within the cytoplasm. Compatible solute play a role in plant osmotolerance by various ways, protecting enzyme from denaturation, stabilizing membrane or macromolecules, or playing adaptive role in mediating osmotic adjustment. The function of compatible solute is not limited to osmotic balance. Compatible solutes are typically hydrophilic and be able to replace water at the surface of protein or membranes, thus acting as low molecular weight chaperones. This solute also function to protect cellular structure through scavenging ROS. Compatible solutes are small molecule, water-soluble, and uniformly neutral with respect to the perturbation of cellular function, even when present at high concentration. They comprise a nitrogen-containing compounds such as amino acid, amines, and betaines, but also organic acid, sugar, and polyols. The three main mechanisms of salinity tolerance in a crop plant: tissue tolerance, where high salt concentrations are found in leaf but are compartmentalized at the cellular and intracellular level, especially in the vacuole, at the process involving ion transporter, proton pumps, and synthesis of compatible solute. Osmotic tolerance, which is related to minimizing the effects on the reduction of shoot growth, and may be related to as yet unknown sensing and signaling mechanisms. Ion exclusion, where Na+ and Cl- transport process predominantly in roots prevent the accumulation of toxic concentration of Na+ and Cl- within leaf. Mechanism may include retrieval of Na+ from the xylem, compartmentation of ions in vacuole of cortical cell, or efflux of ion back to the soil. Interesting facts. Do you know that mangrove trees are one example of mechanism of salt tolerance in plants? The salt glands of some mangrove plants remove excess salt using ion transporter that have a correct concentrated sodium solutions. Salt crystal on Echeveria marina, aka black mangrove, on the leaf. In conclusion, salinity is a significant problem affecting agriculture worldwide and is predicted to become a larger problem in the coming decades. The detrimental effect of high salinity on plants can be observed at whole plant level in terms of plant death or decrease in productivity. Some plant species are clearly more flexible than others in their requirement for survival in salty environments. An understanding of how single-cell responses to salt are coordinated with organisms and whole plant responses to maintain an optimal balance between salt uptake and compartmentation is fundamental to our knowledge of how plants successfully adapt to salt stress. Okay, that's all from me. Thank you for watching.